Hello class! Today we begin Chapter 5, Exploring Mixtures and Their Separation. Mixtures are everywhere, in food, drinks, medicines, factories, nature, and even inside our bodies. In this chapter, we will discover how mixtures are formed, how they behave, and how science helps us separate them. Look closely. The white sugar you enjoy every day begins its journey as green Cbse juice. To get those clean, sparkling sugar crystals, we carefully separate the unwanted materials step by step. This separation science, working quietly in the real world, from a huge factory all the way to your dining table.
Chapter 5 is titled, Exploring Mixtures and Their Separation. All right, class, first thing, the name of our chapter is Exploring Mixtures and Their Separation. So the whole story is about two things, what mixtures are, and how we pull them apart. Everyday activities, like getting white sugar crystals from green sugarcane, rely on the science of separating mixtures. Think about the white sugar you eat. It starts as green sugarcane juice. Getting clean white crystals from that green juice is nothing but separation science working in a factory. So this topic is not just in books. It's on your dining table. Doctors can detect diseases like malaria using just a few drops of blood, thanks to separation techniques. Here's something amazing. Doctors can catch a disease like malaria from just a few drops of your blood. How? By separating and studying the parts of the blood. See how useful separation is? This chapter explores mixtures in greater depth, their properties, behavior, and the techniques used to separate them. In this chapter, we'll go deeper than before. We'll study what mixtures are made of, how they behave, and the clever tricks used to separate them. Separation of mixtures matters everywhere, from industrial sugar production to life-saving medical tests. Remember this line, children? Separation matters everywhere. From a huge sugar factory to a tiny, life-saving medical test in a hospital. Think it over. Why do suspended particles settle in muddy water over time, but not in milk? Question for your brain. If you leave muddy water, the dirt slowly settles at the bottom, but in milk, nothing settles. Why the difference? Hint. It's about the size of the particles. Keep this in mind. We'll answer it later. Think it over. How is evaporation different from boiling? Another thinker. Boiling and evaporation both turn liquid into vapor, so are they the same? No. Boiling happens fast at one fixed temperature. Evaporation is slow and happens at any temperature, from the surface only. Think it over. Why do you see bright rays of sunlight when it passes through small gaps between the leaves of a dense tree? Ever noticed bright rays of sunlight coming through gaps in the leaves of a thick tree? Lovely, right? There's real science behind why we can see those rays. We'll uncover it at the end of the chapter. The Tyndall effect. 5.1. How can we classify mixtures? A mixture of sugar and water has a uniform composition throughout. Take sugar dissolved in water. Every part of it is the same. Top, middle, bottom, all equally sugary. We call that uniform composition. 5.1. How can we classify mixtures? A well-stirred sugar-water mixture is equally sweet in the first and the last sip. Proof? The first sip and the last sip taste equally sweet. That tells you the sugar is spread evenly everywhere.
Here's something truly amazing. Doctors can detect diseases like malaria using only a few drops of blood. How? By carefully separating and studying the different parts of the blood. Just look at how powerful separation can be. It doesn't only help in factories and kitchens, it helps save human lives every single day.
How can we classify mixtures? Such a uniform mixture is called a homogeneous mixture, also called a solution. A nice uniform mixture like this has a name, a homogeneous mixture. And we also lovingly call it a solution. Write that down. How can we classify mixtures? Another homogeneous mixture is vinegar, which is acetic acid in water. Here is an example you use at home. Vinegar. It is just acetic acid dissolved evenly in water. Homogeneous. How can we classify mixtures? Aerated drinks like soda are also homogeneous mixtures. Carbon dioxide dissolved in water. Soda, or any fizzy drink, is carbon dioxide gas dissolved in water. Again, homogeneous. How can we classify mixtures? A solution always remains homogeneous. Key point: a solution stays homogeneous. It does not separate into layers on its own. How can we classify mixtures? A stirred mixture of sand and water is not uniform. Now the opposite. Stir sand into water. Is it uniform? No, the sand does not spread evenly. How can we classify mixtures? In sand and water, the sand particles are easily visible and settle with time. You can actually see the sand grains, and if you wait, they sink to the bottom. That is a big clue. How can we classify mixtures? Such a non-uniform mixture is called a heterogeneous mixture. Homo means same, and hetero means different. That is an easy way to remember it. Ponder this. Is a mixture of oil and water homogeneous or heterogeneous? Think about it. Oil and water do not mix. They form layers. So what do you think? Ponder this. Can you think of other heterogeneous mixtures? Can you list some other heterogeneous mixtures from your kitchen or surroundings? Try before the next class. Activity 5.1. This is a group activity. Divide the class into 3 groups: Group A, Group B, and Group C. For this activity, we will split the class into 3 teams. Teamwork time.
Dilute. As ingredients, it's misconcentrated. Same ingredients, different strength, that's concentration.
Activity 5.1. Each group prepares a mixture in a colorless, transparent tumbler or a 100 milliliter beaker. Each group takes a clear, see-through glass or a small beaker to make their mixture. Activity 5.1. Group A. Add 1 spatula of common salt to 50 milliliters water and stir well, then label it A. Group A. Add 1 spatula of common salt to 50 milliliter water, stir well, and label your beaker A. Activity 5.1. Group B. Add 1 spatula of chalk powder to 50 milliliters water and stir well, then label it B. Group B. Add 1 spatula of chalk powder to 50 milliliters water, stir well, and label it B. Activity 5.1. Group C. Add a few drops of milk to 50 milliliter water and stir well, then label it C. Group C. Add a few drops of milk to 50 milliliters water, stir well, and label it C. Activity 5.1. Observe whether the particles are visible in each mixture and record it. Observe carefully. Can you see the particles in each one? Write down what your eyes tell you. Activity 5.1. Direct a laser pointer through each beaker and observe from the side, perpendicular to the beam. Shine a laser pointer through each beaker and look from the side. Watch whether you can see the beam's path inside. Safety first. Never look directly into the laser beam. It can cause irreversible eye damage. This is a very important safety rule. Never ever look straight into the laser. It can permanently damage your eyes. Point it away from faces. Activity 5.1. Predict what you would observe if the beakers were left undisturbed for a few minutes. Before you wait, predict. What do you think will happen in each beaker if you leave them still for a few minutes? Activity 5.1. Set up a filtration apparatus and filter each mixture. Check for residue on the filter paper. Filter each mixture through filter paper and check. Did anything get left behind on the paper? Activity 5.1. Decide, from your observations, whether these are the same or different types of mixtures. From all your observations, decide, are these 3 the same kind of mixture or different kinds?
Hello, class. Today we begin Chapter 5, Exploring Mixtures and Their Separation. Mixtures are everywhere, in the food we eat, the drinks we love, the factories that build our world, and even inside our own bodies and bodies. In this chapter, we'll discover how mixtures are formed, how they behave, and the amazing ways science helps us separate them. So get ready, because by the end, you'll never look at a simple glass of water the same way again.
Activity 5.1. These are indeed different types of mixtures. Salt made a solution, chalk made a suspension, and milk made a colloid. Hold on to these words. We will meet them properly soon. Solutions. Solutions are homogeneous mixtures. Let us start simple. Every solution is a homogeneous mixture. Same everywhere. Got it? Solutions. A solution forms when a solute is mixed with a solvent. A solution is born when we mix a solute into a solvent. Two new words. Let me explain each. Solutions. The solute is the substance that gets dissolved. The solute is the thing that dissolves and seems to disappear, like the sugar. Solutions. The solvent is the substance that dissolves the solute. The solvent is the thing that does the dissolving, usually the larger amount, like the water. Solutions. In sugar and water, sugar is the solute and water is the solvent. So in sugar water, sugar is the solute and water is the solvent. A small helper trick. The solute is usually the smaller quantity. Solutions. In what proportion are the solute and solvent present, and can this be expressed quantitatively? A fair question. In what proportion are the solute and solvent mixed, and can we put a number on it? Yes, we can. That is coming next. Concentration of a solution. ORS, or oral rehydration solution, is made by adding specified amounts of salt and sugar to a fixed amount of water. Take ORS, the drink doctors give when you are dehydrated. It is made by adding exact amounts of salt and sugar to a fixed amount of water. Concentration of a solution. Changing the amount of salt or sugar, or the water volume, gives a solution, but not ORS. If you change those amounts, you still get a solution, but it is no longer proper ORS. Concentration of a solution. You cannot freely add any amount of salt and sugar to a fixed amount of water and still call it ORS. So you can't just throw in any amount of salt and sugar and call it ORS. The amounts matter. Concentration of a solution. Not all sugary drinks made at home or sold in the market are ORS. Warning children. Not every sweet drink at home or in the shop is ORS. Don't get fooled. Concentration of a solution. Farmers must mix the right amount of pesticide with a fixed amount of water. Here is a real-life example. A farmer must mix the right amount of pesticide with a fixed amount of water. Concentration of a solution. Too little pesticide may not protect crops. Too much can damage crops, soil, and the environment. Too little pesticide and crops stay unprotected. Too much, and it harms the crops, the soil, and nature. Balance is everything. Concentration of a solution. The right proportion is always essential when preparing a solution. So remember the golden rule. The right proportion always matters when making a solution. Concentration of a solution. Concentration is the amount of solute dissolved in a given amount of solvent or solution. This idea has a name: concentration. How much solute is dissolved in a given amount of solvent or solution? Concentration of a solution. Concentration matters in medicine, agriculture, food, and cosmetics, even in making a cup of tea. Too much tea leaves and it turns bitter, right? Meet a scientist. Dilip Mahalanobis was an Indian pediatrician. Let me introduce a hero. Dilip Mahalanobis, an Indian children's doctor. Meet a scientist. He first developed and implemented treatment for dehydration caused by diseases like diarrhea and cholera. He was the first to develop and actually use a treatment for the dangerous dehydration caused by diarrhea and cholera. Meet a scientist. He formulated the ORS that revolutionized rehydration therapy. He created the ORS formula that completely changed how we treat dehydration. Meet a scientist. ORS has saved millions of lives after the WHO popularized it worldwide. After the WHO spread it worldwide, ORS has saved millions of lives. A simple solution? A giant impact. Expressing concentration. Concentration can be expressed in several ways. A common one is percentage. There are several ways to show concentration. The most common is as a percentage. Expressing concentration. There are 3 main percentage methods to express concentration. We will learn 3 percentage methods. Don't worry, they are all cousins of the same idea. Expressing concentration. Collect commercial packaged products and note the composition information on each. Here is a little task. Collect some packaged products from home and read the composition printed on them. You will spot these percentages. Mass by mass percentage. Mass by mass percentage is commonly used for homogeneous mixtures. First type, mass by mass. We use it a lot for homogeneous mixtures. Mass by mass percentage. It tells how many grams of solute are present in 100 grams of total solution. It answers a question. How many grams of solute are in 100 grams of the whole solution? Mass by mass percentage. The formula is mass by mass percent equals mass of solute divided by mass of solution times 100. Note it down carefully. Mass by mass percentage. It is also used for heterogeneous mixtures, like milk powder and spice mixtures. It is used for heterogeneous mixtures too, like milk powder and spice mixes. Mass by mass percentage. It is used to label the composition of packaged foods, like salt, sugar, and protein content. It is what companies print on food packets. Salt content, sugar content, protein content, and so on. Example 5.1. 10 grams of salt is dissolved in 90 grams of water. Suppose we dissolve 10 grams of salt in 90 grams of water. Example 5.1. Total mass of solution equals 10 grams plus 90 grams, which is 100 grams. Solute plus solvent equals solution. Example 5.1. Mass by mass percent equals 10 divided by 100 times 100, which is 10%. So, mass by mass percent is 10%.
And that brings us to the end of this part. Well done, dear students. You have taken one big step in understanding mixtures and their separate, so don't miss it. If you enjoyed this lesson and it helped you, please subscribe, like this video, drop a comment, and share with your friends who are also studying. Keep learning, stay curious, and I'll see you in the next part.
3 simple ways to measure how much solvent is hidden inside a solution. Mass M volume percent, tilde meng MV percent, and going to mass percent. MV percent means is hidden inside a solution. 3 simple ways to measure how much solvent is hidden inside a solution. Mass, the volume percent, volumed incleus for volumed, a first, and a volume per volume mushier in salient. Keep adding sugar, and eventually, no more will dissolve. That's a saturated solution. Heat helps solid dissolve, but it drives gases out. During the vinegar si sforzio entisent, the stunned, it drives gases a scout. As it cools, pure crystals grow. This is crystallization. Boil the liquid, then cool the vapor back down. That's distillation.